right, well, thanks for coming and for having me here. Thanks for the invitation to talk to you today. I'm also so I'm going to echo what Diana said this morning about being a little nervous about this topic to speak with you. So Diana talked about this topic this morning about the competition, the word that no one wants to talk about between wild bees and honeybees. What I'm going to talk to you, at least at the end of this talk today, is about disease spillover between managed bees and wild bees. But before we get there, we're going to talk a whole bunch about transmission of diseases, which has been probably on everyone's mind over the past few years. And we're going to talk about disease transmission in pollinators, okay? So I am not the person who did almost any of this work. All of the people who did the work I'm showing you right here. So Paige Muniz is my lab manager. She's a native bee systematics expert and also a great beekeeper too. Caitlin Deutsch is a PhD student. So Caitlin is actually from Gainesville, Florida. She grew up right here in the state of Florida as a beekeeper, and then I attracted her up to New York. Laura Figueroa is a former PhD student, now a new faculty member well, we're seeing where she becomes, she's being recruited by a few different places. And Pete Greystock is a former postdoc, now a faculty member at Imperial College London. In addition to that, we've been having a lot of fun with this topic over the last, I think it's been about 7 years since we've been funded on this topic from various federal agencies. And the team is really fun. So we have a theoretical ecologist, Steve Elner, and a network modeler, Chris Myers. Both of them are at Cornell with me. And then we have 3 bee people as well. So Lynn Adler, Becky Irwin, and Quinn McFrederick. Okay, so I'm going to tell you a little bit about some of the data that we've collected over the past, about 7 years or so on this particular project. All right. I thought that I was not going to have a lot of time, but apparently I'm starting pretty early, so I do have some time to talk to you about this. I'm not even going to do much of an introduction. I'm just going to blow right into it. So why are pollinators having a lot of problems? I'm sure you've seen various slides like this before. This is my slide for this particular thing. We know it's not any single factor for why honeybees are not doing so great and why other pollinators are not doing so great right now. It includes pesticides or agrochemicals, pests and pathogens, climate change, habitat or the food that the bees are eating and the places they're living, and also when they're inadequate at management practices. My lab studies 3 of these in our primary goal. We study a little bit about habitat and climate change, but these are the 3 main things that we study in our lab. Pesticides, pests and pathogens, and also management practices. I'm not going to talk about pesticides today, but what I did want to let everyone know who's in the room here is I've been getting very involved in this topic and providing resources for this topic for about the past 4 years now. So in addition to being a professor at Cornell, I'm also the director of our chemical ecology core facility. Our manager is shown right here, so this is Wayne Anderson. He has a PhD in chemistry. He's a much better chemist than I am. But what we offer is a 93 pesticide multi-residue analysis. That's open to anyone in the public. If you're interested in testing your bees or your pollen or your honey or the wax comb that you have or any of the plants around your apiaries, you can send us a sample. It's $90 a sample. That's our at cost. We can't make a profit. We can't lose money on it. So that's what covers the cost right now. And we process about 1,500 samples a year. Just to give you a picture of who's using this right now, we do the Bee Informed Partnership samples. We do Fish and Wildlife Service, the U.S. Fish and Wildlife Service, several USDA labs, the Canadian Agriculture and Agri-Food Canada, Xerces Society for Invertebrate Conservation, a whole host of government and non-government organizations, lots of research projects and universities. And then what I'm trying to promote, why I'm telling you about this today, is I'm trying to promote this to hopefully be broader used by beekeepers, farmers, and members of the public. I think that the more data, me being a scientist, I guess you can see right through me, right? More data means more knowledge. So we've been working with 62 beekeepers over the last 3 years, 37 farmers, and 9 homeowners. If you'd like to see what's in your samples, send us a sample, okay? So I'm easily Googleable. Just Google Scott McArt. You can also search for us, just do Cornell pesticide analysis. We're the first thing that comes up on a Google search, or that's our website right there, blogs.cornell.edu, CCECF, so for the Cornell Chemical Ecology Core Facility. All right? Any questions on that before I get going?
How do the bees have to be sent?
So email me. Depending on your question, there's a lot of different ways to prepare a sample and different ways to collect a sample. And the best way to get the answers that you're looking for is for me to interact with you or for Wayne to interact with you. Okay? So just give us an email. Sorry, what? Previous, I might be able to. Is it this button? Yes, okay, good. So turnaround time, we had a machine breakdown over the last summer. So now our turnaround time is around 4 months. I realize that that is not a good turnaround time. So I am currently in the process, well, we're playing a lot of catch up right now, and I'm currently in the process of trying to hire one more person. So if you know anyone with a lot of money who wants to help me hire another person, let me know. But that's what I'm doing in the background right now, is trying to get that so we can hopefully have her turnaround time be more along the lines of a month or two as opposed to 4 months. Okay, any other questions? All right, it's actually really hard to see with the lights beaming down on me here. Okay, so past pesticides, now let's talk about disease. All right, so we'll focus mostly on pathogen transmission among bees, so for the first two things I'll talk about, and then we're going to talk about how management practices, especially among beekeepers, can influence disease in the entire community at the end. So here's the outline. First, we're going to talk about how diseases are transmitted among bees. And we're not going to focus on in the hive. There's been a lot of work that's actually gone on disease transmission within hives. But we're going to focus on where there's not been a lot of work, and that's transmission out in the environment. Okay? Then we're going to focus on, is there seasonality of bee diseases? Anybody in here know anyone who's had flu lately? Had the flu? Almost everyone, I'm guessing, right? If I had asked this same question in the summer, do you think you would say the same thing? Probably not. There's seasonality to human diseases. So is there seasonality for bee diseases? We're going to talk about that. And then finally, how can we limit disease transmission and spillover, right? So at the end of the day, once we understand the system, then we want to do something to make everyone healthy, or make our bees healthy in this circumstance, right? So we'll finish up with that topic. All right, so we'll start out. How are diseases transmitted among bees? One of the main places where pollinators interact out in the environment, either in space or in time, are at flowers, right? So you can see these two pictures right here. There's a honeybee and a bumblebee on the flower at the same time, and the other one, there's a honeybee and a little sweat bee on the flower at the same time. Sometimes that occurs, but sometimes it's not exactly, at the same time that those bees are visiting. There might be one pollinator that visits the flower, and then a little bit later, a different pollinator visits. But they're still sharing that flower, right? And still potentially transmitting diseases when they're either there together, or if they're there in sequence, if the disease can actually, or if the parasite or pathogen can hang out on the flower long enough to be able to be transmitted. So our question was, can flowers be these dirty doorknobs, right? So in the kindergarten classroom, you go in, you open the door and you say, maybe I should use some hand sanitizer after that, right? So could flowers be these dirty doorknobs that facilitate disease transmission? All right. So how might this work? Just to really hammer this home. Let's say a honeybee with these 3 green blobs, these 3 pathogens inside of the honeybee right there. It comes along, forages at a flower. It deposits those pathogen particles. Maybe it defecates on the flower, or somehow the pathogen gets rubbed off on the flower. Those pathogen particles are now there on the flower. A different bee, maybe of a different bee species, comes along, picks up those pathogen particles, and if it picks them up and goes back to its nest now and develops an infection, that's successful transmission and successful disease amplification that might occur in that next pollinator, okay? So really, when you break this down, this process of transmission, there's three key components. One is, can the pollinator deposit a pathogen on flowers? So deposition on flowers, number one here. Can that pathogen remain viable on those flowers? So viability on flowers is a second component. If it immediately dies, then no transmission happens, right? But if it can remain viable until the next pollinator comes along, maybe it can be transmitted. And then, of course, it has to be acquired by the next pollinator that comes along and then established in that new host, okay? So what we set out when we first started this topic was to say, well, all right, how likely is it that each one of these factors that's important for transmission actually occurs on flowers? So we'll start out with deposition. At this time when we had just received funding from NIH, actually, that was interested in funding us on this topic, I had a PhD student starting, Laura Figueroa, very talented PhD student, really fun to work with, and she said, I'm really interested in bee poop. So I said, all right, well, let's study a fecal oral pathogen, sort of like, dysentery in humans. There's various things that cause us to have diarrhea. Well, it turns out there's things that cause bees to have diarrhea as well. Sorry, we're getting a little gruesome here after lunch, but it's important for disease transmission. So I said, all right, well, maybe a good model system would be to study one of these fecally orally transmitted pathogens, and a good one is Crithidia bombi. So this is a trypanosome pathogen. You can see it sort of down in your lower left-hand corner here. It sort of looks like a balloon blob with a tail. And it's a known pathogen of bumblebees, but we've been finding, and we knew sort of up until this point, that it actually infects a lot of other bee species as well, including honeybees, okay? So the question was, do bees defecate on flowers? And what we decided to do was bring this question into the lab initially and say, all right, we'll set up a whole bunch of cages, we'll inoculate bees with Crithidia, or have some bees that don't have Crithidia, and then we'll put some fluorescent dye into their food as well. Why is that important? Well, you can see here, if we put black light on the flowers, it's really easy to tell if bees have defecated on the flowers because the dye goes right through them, okay? So we fed them the fluorescent dye, we let them do their thing foraging on these flowers, we watched a whole bunch of these cages for a couple years actually, we watched these cages. And what did we find? Yes, bees actually defecate on flowers quite a bit. So there's some differences here, as you can see, whether a bee is infected or uninfected. The infected bees defecate a little bit more, so kind of consistent with this theme of it giving bees diarrhea, being a pretty smart pathogen, right, to be transmitted fecal orally. If you can cause your host to defecate, you can be transmitted more. But the important point here is just the level of defecation that's actually occurring on the y-axis. So across all of the bees that were put in here, about 2/3 of them defecated on flowers. That's a lot of pooping on flowers, right? And potentially, facilitating transmission. Okay, so that's deposition. I would say, pretty good evidence that we have, or to suggest that, yeah, deposition of a pathogen occurs quite a bit on flowers, at least in a lab setting. What about viability? Okay, so how long are these pathogens actually viable on flowers? To look at this, we went out into the field, so just outside of the lab, not too far into the field, and we put a whole bunch of plants either in the shade, so in sort of a open greenhouse setting, or just open sun. And then what we did is we shook a lot of bees in little tubes and got them to defecate, collected their feces, and then we inoculated the flowers with that feces, okay? So this is already a pathogen that has passed through a bee. Now we put it into, collected all of it in a little tube and put it on the flowers, okay? On average, we know that it's about 10 microliters, so about 5 to 10 microliters that bees will defecate on flowers, because we watched a lot of bees defecate on flowers. So that's what we put on the flowers, is about 10 microliters. Okay, what did we find? We found this. So really, it didn't matter too much. You can see there's some significant differences in survival here. So on the y-axis, you're looking at percent surviving. If it's right at the top at the beginning of time, it's 100% survival. And then survival starts to drop off of Crithidia after some time. And there's some differences among the different types of flowers here. There's some differences in terms of whether you're if we inoculated the inside or the bract of a flower. And there's some differences in whether, the flower was, just in the full sun versus it being in that glass house setting with some shade. But really the important point here is what's on the x-axis. It's around 1 to 3 hours. So a pathogen, at least Crithidia in this case, on these flowers is going to remain viable for about 1 to 3 hours. Plenty of time for another bee to potentially to come along and pick up that pathogen. All right? Okay. So that's viability. We now know that deposition happens, so bees are pooping all over flowers. Kind of makes you think a little bit different about flowers, or at least makes me think a little bit differently about flowers. We also know that at least Crithidia in this setting on a lot of different flowers can remain viable for about 1 to 3 hours. Now let's close the link, all right? So acquisition. Do bees actually come along and can they potentially acquire that pathogen when they forage on a contaminated flower? So to look at this, we went right back into the lab. You can see we put a little bit of inoculum on flowers. That's that little orange dot there that you can see in the picture. And we put it in a lot of different places. So either inside the flower, inside the corolla, outside the corolla, or on the bract of a few different plant species here. What did we find? Well, what we found is about 50% of the time, a bee put into that cage, foraging on that flower, did in fact become sick, so they acquired the pathogen. And sort of interestingly, what I'm showing you here is that infection intensity varies depending on the contamination site. So some of those light blue bars over to the middle and right-hand side of the graph there, what you can see with those light blue bars is actually when you put the pathogen on the bract, so not inside the flower, but where the bee is mostly sort of putting her legs, that's where they seem to be picking up the pathogen the most, all right? So, but again, the important point here is it's about 50% of the time, at least in our lab setting, in these controlled cage trials, that they were picking up Crithidia. Okay, so if we come back to this main question, can transmission occur at flowers? I would say from this that we have pretty good evidence that transmission can indeed occur at flowers, right? But I know what you're saying, and well, you should be saying, because I was saying the same thing, is, all right, Scott, this happens in the lab. What about the field? All right, so again, I'm not going to show, I thought I was going to have a little bit less time than I actually have, so maybe there's some slides at the end we can go to if you guys want to. But I'm not going to show you all the data that we have on this, but I am going to show you what I think is the most important piece of data that at least suggests to me that this probably is fairly common in the field. All right. So to do this, we went out into 3 old fields in New York. Anyone from the Northeast and seen old fields like this before? They're spectacular, right? I know you do. So anyways, old fields, there are big fields in the Northeast. Lots of different bee species that are there that I'll show you in a little bit. And it's also filled with goldenrod in the fall, which this gets my bees through the winter. So I tank up my bees on goldenrod at the end of the season, and that, boy, they're all set with honey for the rest through the winter. So what we did is Pete, my former postdoc, and then Paige, my lab manager, and a whole team of us, we went out and we sampled bees and flowers across the entire field season, so for all the way from May until October, at 3 different old fields, just right around Ithaca, central New York. We screened for 5 pathogens, so now we're expanding this beyond just Crithidia, which I showed you in the lab trials, and now we're going to look at 5 pathogens simultaneously. Overall, we screened almost 3,000 flowers from 89 species and almost 3,000 bees from 110 species. So this was just a massive undertaking where we sampled a ton of stuff, all right? Okay, so what are the pathogens we looked for? We looked for 2 microsporidians. So we did PCRs to look for Nosema bombi and Nosema cerani. Most people in this room you're probably familiar with Nosema cerani. This is the main Nosema species that causes nocemosis in bees. We also looked for a neogregarine, Apicystis, and then 2 trypanosomes. So we looked at Crithidia bombai again, and then also Crithidia exboecki, which is a little bit more abundant in honeybees and some other bees. All right? What did we find? All right, so again, I'm not going to show you all the bee data, but I will show you the flowers, because I think the flowers, personally, I think this is maybe the most interesting result I've seen in the last 5 years or so. But I'm totally biased because obviously this is coming out of my lab. Anyways, I wanted to show you the flowers because that's also, I think, a key to whether transmission could actually occur. If we see a lot of pathogens on flowers, all right, yeah, maybe transmission happens fairly frequently. If we don't see a lot of pathogens on flowers, maybe this is something that we can sort of get to work in the lab, but it doesn't really have much relevance to the field, right? All right, so what do we find with those almost 3,000 flower samples? Turns out bee pathogens are all over flowers, okay? So what I'm showing you here is a heat map of all of those 89 species, 89 species of plants, 110 species of bees, but this is the plant data. So 89 species of plants, right? So that's all that tiny, tiny little text going down on the y-axis there. And then you've got the 5 pathogens, so Nosema bombai, Nosema cerani, Crithidia bombai, Crithidia exboecki, and Apicystis across the x-axis. If you have more color, so more sort of darker green, that means there's a higher prevalence of bee pathogens on those particular flowers, okay? Basically, the take-home point here is that there's a lot of green, okay? There's a lot of bee pathogens out there. 70% of flower species have a bee pathogen on them, and 9% of individual flowers have a bee pathogen on them. That's crazy, right? 1 out of 11 flowers, on average, have a bee pathogen on it. I don't know about you, but I have a lot more than 11 flowers just in the garden in front of my doorstep at my house. And on average, at any given time, over the course of the season, from what these data say is, one of them at least is going to have a bee pathogen on it, okay? I think this is pretty strong evidence combined with the lab-based data from before that pathogen transmission at flowers is probably fairly common, all right? And we can argue about that later if you want, but that's my take on it. Okay, so how are diseases transmitted among bees? I think floral transmission is probably fairly important. Then the next thing we're going to address here is there seasonality in these bee diseases? Again, making this connection to human diseases, which have some seasonality, is there something similar that occurs in bee communities? All right. Anybody recognize this graph? Everyone recognizes this, right? We've all seen this in the newspaper, right? So this is COVID, unfortunately, COVID deaths over the last 2.5 years. Even with COVID, there's seasonality. So winter is a time where you want to be putting that mask on. And I know I'm not being very smart, just like a lot of other people are in this room aren't being very smart. But we should all be wearing masks, right? Because COVID is transmitted in the winter, flu is transmitted in the winter. If we want to reduce transmission, we need to implement some measures about it. That's a whole separate topic. We won't get into that topic, but what we will talk about is diseases of bees. Okay, so here is a graph showing seasonality of a pollinator community in this New York old field, okay? So this is the same data as I was showing before, showing the prevalence of all the different pathogens, but now we're looking at, which bees are abundant at different times throughout the year. And I think the results are pretty intuitive, actually. So on the left-hand side of the graph, we're moving from May to September here, right? So May is the left-hand side, and then September is the right-hand side. In May, the most abundant bees in this community are not honeybees or bumblebees, okay? The most abundant bees are small carpenter bees, Ceratina, that's what's shown in the green here, sweat bees, particularly Lasioglossum, and then other bees, things like mason bees, osmia, leafcutter bees, megachile, et cetera, okay? As you progress through the season, you can see the relative proportion of the bees changes dramatically. So by the end of the season, what do we see a lot of? We see a lot of bumblebees and honeybees. And this makes a lot of sense, right? Bumblebees and honeybees, the colonies start very small, early in the summer, but they build and build and build and build, and then they become the dominant bee by the end. Usually when I'm looking for bees in September and October at my place in New York, it's almost all honeybees, right? It's almost all my honeybees, actually. Okay, so there's a seasonal turnover that you can see here in the bee community. What about bee pathogens? Turns out there's actually a very pronounced seasonality to bee pathogens as well, okay? So what I'm showing you here is that so the x-axis is the same. You're starting in May, and then you're ending up in September, and then all of those different lines are different bee pathogens. The one at the top with the black is the total level of pathogens. And you can see a huge increase, right? So this is nearly a 10-fold increase in bee pathogens from where the start of the season to where they ended at the season, okay? Who is driving that, right? So if we go back to this particular graph, you can see this seasonality, right? And you remember Apis and Bombus are the ones that are very abundant at the end. Well, probably no surprise that if you now look at the contribution of each bee genus to parasite prevalence, to prevalence of these pathogens, you see a huge sliver of orange and red towards the right-hand side of that graph. That means a lot of this increase in disease later in the season is due to honeybees and bumblebees, okay? All right, so there's the seasonality of bee pathogens. Now we'll wrap up with what I think is probably the most important topic of the whole talk here, is how can we limit disease transmissions, all right? So some bees are sick, but if we want to improve the situation, how can we potentially limit disease transmission? And again, I wish I had known I was going to start 15 minutes early, because then I would have told you the story about planting more flowers. But the best way we can do this, without showing you data to support it, the best way we can do this is just to plant more flowers, right? And I think it's fairly intuitive why this would be the case. If you plant more flowers, you have higher numbers of flowers compared to your numbers of bees. They're pooping on those flowers less, or visiting those flowers less, and therefore they're just transmitting less disease in those places. The added benefit of having more flowers is it's more food for bees, right? So it's a win-win scenario. I'm not going to show you data, but that's obviously the best way. All right, now, here we go. Here's the thing I'm nervous to talk about. You also can make sure that your bees stay healthy. Okay, so what happens if a honeybee gets sick in our system? So what I'm showing you here is the entire Plant-Pollinator Network of our old fields in New York. On the left-hand side here are all of the 110 bee species, okay? And on the right-hand side are all the 89 plant species. All those lines between them are where they share interactions at flowers, okay? So the question is, if this honeybee gets sick, can it potentially spill over all of the diseases to those other pollinators? The opposite could be true as well, right? So it could be other pollinators spilling over their diseases into honeybees, or spill back into honeybees. But because we know from our data that the honeybees and the bumblebees especially are the ones that have the most pathogens at the end of the season, we think that it's probably direction for both honeybees and bumblebees here, that they're the ones that are probably passing it to the other bee species, okay? So what is the likelihood that this actually occurs towards the end of the season? To look into this, we worked with the New York State Beekeeper Tech Team. This is a great state-funded program. I wish every state had one of these. I feel very fortunate to be in New York State, where the governor has put a lot of money behind pollinators, thanks in part to Dan and other people who were very strong advocates for our pollinator protection plan in New York. But anyway, so we've been working with beekeepers in a very applied sense since 2016. And one of the main things, not surprising to anyone in this room, one of the main problems is Varroa management, right? So if you control Varroa, your bees do relatively well. If you're not controlling Varroa, they don't do well. Not rocket science, right? Every beekeeper knows that. Hopefully everyone in this room knows that. It's not necessarily Varroa that causes the problems in your bees. It's the viruses that they transmit, okay? And I think Diana's going to talk to us a whole bunch about these viruses tomorrow or well, she knows a lot more than me on the topic of viruses and honeybees. But so anyways, it's deformed wing virus, Israeli acute paralysis virus, various of these viruses. They're the things that really kill the bees. Varroa just makes it possible by weakening the immune system and also transmitting these viruses. So Caitlin Deutsch, our beekeeper from Gainesville, Florida, who's been doing a PhD in my lab, became very interested in this topic of wondering, okay, if beekeepers are not managing Varroa well, their virus levels are probably high in their apiaries. What I've just showed you with other pathogens is those pathogens can spill over onto flowers. What about the flowers surrounding apiaries for when Varroa is not controlled very well? Are those filled with viruses, and are other bee species picking up those viruses? So we worked with the tech team. We worked with 27 apiaries this particular year. These are the Varroa levels. So across the x-axis here is the colony level Varroa load, average colony level Varroa load, your mites per 100 bees across all these 27 apiaries. As you can see here, some people were controlling Varroa very well. That's on the left-hand side of the graph. The 3% threshold is that with that red dotted line. So some people were controlling Varroa very well, and about half of beekeepers were actually above threshold, so above 3% in September. Again, this main time when we know honeybees are contributing a lot to pathogen prevalence in the whole bee community, all right? Okay, so what did Caitlin do? She went out and collected bumblebees surrounding all of these apiaries and screened them for DWV, so deformed wing virus. What I'm showing you here is a graph. On the x-axis is your average Varroa load per 100 bees at each apiary. So each point on this graph is going to be one apiary. And then on the y-axis is the DWV prevalence in bumblebees. So she screened 20 bumblebees at each site. A higher proportion of bumblebees that were positive for DWV will mean that the level is higher, okay? So if you have a positive relationship here, that means there's probably some sharing of deformed wing virus that's occurring. And that's exactly what we find, okay? So as average Varroa load goes up at the apiary level, DWV prevalence in wild bumblebees surrounding those apiaries also goes up, okay? And I know I'm nervous to say this. But here's the question, right? What impact is that DWV actually having on wild bees? Is it just some bits of RNA that are floating around in those bumblebees and other bees? Or is it actually having an impact on them? Are the bumblebees getting sick, and are they dying quicker? So to start looking into this, and I will definitely say this is a start at looking into this because we need to do more work on this. But we started by injecting some of these other bees. So we injected the common eastern bumblebee, the Bombus impatiens, and then the two other bees that are commercially available. So we got horn-faced bees, Osmia cornifrons, and the blue orchard bee, Osmia lignaria. And we started injecting these bees with a DWV strain that was sourced from honeybees, and at levels, we injected them with levels that are known to cause infections in honeybees. And we said, all right, what's going to happen to these other bee species? Are they actually going to get sick, and is it going to have some impact on their survival? And you can see, unfortunately, it does. Okay, so bees that were injected with DWV are shown here in blue, and bees that were injected with just a control phosphate buffered saline, just as sort of a buffer, but just to mimic, exactly the trauma that occurs with a stab, in each of these circumstances. They're shown in red, okay? On average, so you can see survival, right, over survival days, it goes up to 50 here, and then the proportion surviving gets lower and lower as you go out here. What you can see is that bumblebees, Bombus impatiens, dies about 3 times faster when it's injected with DWV compared to the controls. How about another bee? So this is horn-faced mason bees. They die about 2 times faster, right? So definitely getting sick from this injection. And then what I think is very actually indicative of our other work, not virus work, but other work on other pathogens, is it varies by bee species. So here's Osmia lignaria, the blue orchard bee, and there's no impact of DWV. I don't think this is particularly surprising, right? So COVID, for example, just to make the COVID analogy throughout this entire talk, COVID probably came from a different animal, probably bats. It made the host switch into humans, and it also infects some other mammalian species as well. But it doesn't infect every mammal, right? And it's worse in some mammals than others. So I don't think it's very surprising that DWV should be the same. It impacts some bee species, but not others, right? Okay. So what are sort of the overall conclusions from what I've told you? Well, some bees are impacted, others are not. We've just said that. I think, and this is why I think we're early in this particular line of research, we need to understand oral transmission. It's almost impossible that a flower is going to stab a bee with DWV. So how important is that, right? So I can show it in a lab setting that this has impacts on bees. Well, what about the real world? In the real world, bees are probably picking up small amounts of DWV from flowers, mostly through their mouths when they groom, would be my guess. So we need to understand oral transmission, which is probably much more realistic than these injections, okay? And then I think, maybe the most important thing that I can say to this room on this topic is there is always a tension between agriculture and non-target organisms, right? I do a lot of research with pesticides right now. I talk to farmers, and I talk to beekeepers, I talk to conservation groups, I talk to various audiences, and there will always be a tension between pesticides and non-target organisms. At the end of the day, farmers need to use pesticides. It's just the way our agricultural system is set up. The key is how to do this sustainably, right? How do you minimize impacts on non-target organisms? Because pesticides aren't going away. The same is true on this topic, right? Beekeepers, in this sense right now, is agriculture, right? So we're producing honey, we are providing pollination services, et cetera. Beekeeping is agriculture. The non-target organisms are these wild bee species. There will always be this tension, as Diana alluded to regarding forage as well. This is a very controversial topic. The reason it's controversial is because we don't have a lot of data and we don't know how to make things sustainable. Hopefully, by providing more data on this topic, we will be able to increase sustainability of this system. And honestly, aside from planting flowers, which is by far the best thing that we can do, it's pretty easy, right? If you control Varroa, you're probably not only helping your own operation, but you're also probably helping the entire bee community, at least the bees that can potentially have transmission of these pathogens and get sick from them, okay? All right, so I'll get down off my soapbox now. And I'll just say thanks for your attention. Again, this is on the non-disease transmission topic on pesticides, if you guys want to, please send me a sample. We'll communicate results with you. Happy to talk about all sorts of things regarding pesticides. I write a monthly column for American Bee Journal. I'm always looking for people to tell me what they want to read about. So if you read this or if you're interested in this, tell me what topics you want to hear more about, and I'll try to highlight those particular papers. And then lastly, just thanks to NIH and USDA, who have both been providing a lot of great funding on this, I know, fairly controversial topic, at least the second part of this talk being fairly controversial. So very appreciative to them, all the people in my lab who helped out with this, and then Ellen up there, I just want to point her out especially. So Ellen Topitzofer, we've just stolen her from Oregon State University, and she's coming to Cornell. She's going to be our new honeybee extension associate, and she is awesome. So hopefully we'll get Ellen down to ABF soon. Okay, thanks very much, and happy to take questions. If we have time for questions.
Yes.
We have time for questions. Okay, great.
You only have 32 minutes for questions.
Oh, this is dangerous. Now I've just given a controversial topic talk, and I've got 32 minutes for questions. The tomatoes are going to come out.
Yeah, okay, so I have a question. I'm assuming that one test is not really going to show us a lot because we know we have viruses in our bees. So when is the best time to test, or like I have 14 different bee yards. I have most of them in a very densely populated area of Chicago, and a few of them in a very remote area. Should I test the densely populated at the same time as I test the remote in late summer? Is that a better time?
Yeah, and so you're talking about testing for viruses?
Yes.
Is that correct? Yeah. So there is a seasonality to viruses. We know that some viruses track Varroa fairly well. So as Varroa goes up, some of the ones that are either transmitted by Varroa or become amplified by Varroa go up as well. So in general, what I would say is, a good sort of rule of thumb, this is not going to be true in every circumstance, but certainly could be in the majority of circumstances, is to look for viruses. I mean, if you're wondering, if the question is when are they most likely to be spilling over into other bees or impacting your own bees, a good time to look is right after Varroa peaks. So typically around September or October in most regions of the country. That's not going to be true all the time. There is not strong seasonality, for some viruses that could potentially be transmitted. And I guess the other thing to consider too is, do you want to wait until you're already having a potential problem, or do you want to be proactive about it? If you want to be proactive about it, maybe it's better to, try to catch it before things really go up. That's certainly what we do for Varroa, right? So we're always trying to get, find it before it goes above threshold and then treat. So if that's the case, maybe you should be looking every month if you have the funds.
Yeah, thank you.
Yep.
Thank you very much for that presentation. My question was just about the transmission of pathogens and deformed wing virus to these other bee species. Just to use Bombus impatiens as the example, that's my kind of hometown bumblebee as well. Are you just testing wild field bees? And if you were to test manufactured kind of boxed Bombus impatiens that are sold for pollination services, do you think the result would be even more prevalent?
Sure, yeah. So there's actually been quite a bit of work on the topic of diseases in managed bumblebees. Actually, Pete Graystock, the postdoc in my lab, who I highlighted in some of this work, he's actually published some of the major papers on that topic. In the early sort of 2010s, sort of late 2000s and right around 2010 or so, it became clear that some of those managed colonies were actually pretty riddled with viruses and other pathogens. Pete published a paper, it was actually called the Trojan Hives, which is sort of, eerie, right? So it's the Trojan hives. You're putting these pollination or bumblebees out for pollination, but they're potentially transmitting all sorts of diseases. That has gotten a lot better as a result of Pete's work on this topic and as a result of the two companies, Copert and BioBest, really doing a great job screening for pathogens in their operations. They've engaged several scientists. Diana may have been engaged in this. There's a whole host of scientists that have been engaged by those companies to make sure that they have clean stock, that what they're selling is not a Trojan hive. It doesn't have a whole bunch of pathogens in it, but it's actually fairly good. I have not seen any data recently showing what is the prevalence of pathogens in those hives compared to wild pollinators, but my guess is it's probably about the same. There's going to be disease. There's probably disease in this room right now, right? Most people are healthy, but there's going to be a small level of disease pretty much anywhere you go. My guess is, and again, based off of no data, but my guess is it's probably about the same in the managed pollinators and wild pollinators right now.
Thank you. Yeah, I'll follow up with you to get my hands on that research. Thank you.
Okay, sounds great.
Thank
Hello.
you. I understand bees poo and poo on flowers, but in my mind, I am seeing the bees going to the very same apple blossom over and over again, and how they are cued in to repeat going to those flowers. Could their poo attract bees to those flowers, or their honing devices, or what is attracting them to repeat those flowers, could that also have a viral or a pathogen in it, a viral load?
Well, that's an interesting topic. I don't, there's been no work done, to my knowledge, on that topic. You know, if a bee defecates on a flower, does it make that flower either more or less attractive for another bee? It's a fascinating question. I guess my intuition would be it probably makes it less attractive. You know, I'm just thinking of some of the fecal oral pathogens in the human world. I've tried to avoid those myself. But yeah, no, a more serious answer is we just don't know. Yeah, no, I don't know how that would change the attractiveness of a flower or not.
I was just going to ask real fast, are you guys coming up with, or do you know of more natural ways to treat Varroa other than like the Mite Away quick strips are what I've been using twice a year, but do you recommend anything else?
Oh boy, this is a loaded topic in this room. There are many ways to treat Varroa. Max is great. Mite Away quick strips are great. Formic Pro is great. Amitraz is still great. There's resistance, you know, developing to Amitraz. I think Frank Rinkovich is here and can tell you all about that. You know, oxalic can also be great. I've had here's where we start getting controversial in other ways on this talk. I've had less success with other treatments myself. Those are really sort of the three standbys for me. But there's lots of other products that are out there, and there's lots of other products that are in the pipeline right now, in the research and development pipeline for treating for Varroa. I'm sure almost every single person in this room has a different opinion than me and can also talk about their own sort of pet Varroa treatment that they might be most excited about. But I guess that's my short answer. All right. Oh, one more.
Thank you for the presentation. It was wonderful. I heard you mention in your research you measured the infectious particles on the flowers. Did you monitor any disease protective components found on flowers?
We did not. However, that's an active area of research right now in my collaborator Lynn Adler's lab. She has some really cool data showing that plants in the Asteraceae family, so goldenrods, sunflowers, other asters, seem to have anti-parasite activities in bees. There's a new paper that just came out 2 days ago that shows it actually may even reduce Varroa levels in bees. That's actually the next paper I'm going to highlight in ABJ, just because it's crazy, right? Like, oh my God, bees, if you go and eat sunflower, somehow you might be protected. They've looked into the mechanisms of why that might be the case, so why there's this anti-parasite activity in sunflower. They've essentially ruled out chemistry, so it doesn't seem to be chemical compounds like plant secondary compounds. They have not ruled out physical structures yet. So what they've found is that plants or pollen, aster pollen, has a lot of spikes on it. And sunflower actually has huge spikes. Things like goldenrod has kind of tiny spikes on it. But pretty much every aster pollen has some spikes on it. Why spikes would have anti-parasite activity is still unknown. The thought is that it's not an actual physical defense. It's not like impaling Crithidia or Nosema or something like that when it's in the bee gut. The current thought is that it might be stimulating the immune system of bees somehow. How that actually is true, I have no idea, right? That's sort of the state of the knowledge on this particular topic. But what Lynn has shown is that when bees, both honeybees now and bumblebees, consume aster pollen, they're more resistant to Crithidia infections. They can clear Crithidia infections better, and they seem to be more resistant to Varroa, believe it or not. Yeah.
Thank you.
Yeah. Pretty wild, right? Definitely, that is definitely a topic that I hope that there's more work on going forward. It's really neat.
Thank you so much for your talk today, and
Yeah.
thank you for your column in the American Bee Journal. I do read it regularly.
Great, there's one person.
And this topic is particularly interesting to me as a master gardener and a master beekeeper. There's people in my community come to me to ask about, what can I do to save the bees? And when they say, should I become a beekeeper? I'm like, no, plant flowers. So if, you had asked about writing and articles and, what people are interested in, if there was some way to incorporate some layman terminology and making it easy for the layman, it'd be wonderful to be able to hand your articles out to people in our in local areas so that they would say, oh, wow, there's really good proof that we should plant more flowers. So I really appreciate that comment. Thank you.
Sure. Well, thank you for the comment. Yeah, I agree. I give talks all over the place. A lot of bee clubs, big organizations like this, conservation groups, farmers, etc. And I would say the most interest that I've had lately, even from small-scale bee clubs, is this exact topic, right? So how can I make a more pollinator-friendly garden or contribute to more flowers out there? I think there really is, my sense, because of the interest of this, is there's a recognition of what Diana talked about this morning, in that there's less habitat for bees out there. There's just fewer flowers. If you're a commercial beekeeper in the room right now, you know that, right? There's more competition for trying to competition among beekeepers for just trying to find a place to put your bees. But that, I think, is starting to be recognized by the public in some sense. How pervasive that really is in the public, I'm not sure. But I guess, to answer your question, I agree with you that we should exploit that, right? So if there's more interest in the public in this particular topic, we know that planting more flowers is going to just cause all sorts of benefits for bees, whether it's managed bees, wild bees, beauty of nature, etc. Yeah, we should be trying to exploit that and developing more resources and more public interest in the topic. I don't know if that's an answer to your question or just me getting on a soapbox again, but there's my opinion. There's nothing that I'm aware of other than the publication that literally just came out 2 days ago showing that sunflower pollen can reduce, or at least associated with a reduction in Varroa infestation levels. In terms of different flowers that help or either promote or limit disease spread, we actually have pretty good evidence that it varies a lot by flower. We have a couple publications now that I didn't show that sort of group these flowers into particular trait groups, right? I mean, there's thousands of different flower species, and they all grow in different places. So if we try to be species by species in terms of making recommendations, great, that will work for one region, but not another region. What we have found is that flowers that have particular traits, particularly the ones with a more closed corolla, so not open flowers, but closed flowers, and also smaller flowers, transmission is less on those. You actually obviously have to balance that with what sort of nectar and pollen are being produced by those plants. So great, if we want to limit disease transmission, we can try to get one flower, but if it provides no resources for bees, well, what good is it, right? So I wouldn't say we're actively trying this, but we've certainly talked a lot about trying to combine those two things into at least a trait-based recommendation guide. So which particular flowers are limiting disease or which particular types of flowers are limiting disease transmission, and which particular types of flowers in general might be fairly good for providing resources. Yeah, I appreciate the question. Maybe we should put more active effort into that instead of just talking about it. All right, you guys are probably sick of hearing from me, so. All right, thanks very much again.